All right. Now we're talking about epithelial tissue. Epithelial tissue. Okay? The word epi means above. Thus, as I alluded to, epithelial tissue is always on the free surface. Inasmuch as it's on the free surface, it will either line a hollow organ, like the inside of the intestine or the blood vessels, or it covers, like over the skin. However, epithelial tissue is also responsible for making the glands. Glands secrete. We have exocrine glands and endocrine glands. We'll talk about exocrine glands and endocrine glands. So epithelial tissue will do this, but it also does the glands we'll talk about briefly. Okay. The first thing we want to talk about are what are the characteristics of epithelial tissue. They have polarity. Okay, let's say you have a tile floor. If I handed you the tile before it's put down, you would definitely know by looking at it what's the top. It would be the more decorative part, and what's the bottom. It would be the more rough part to go down. So epithelial cells have a definite top and a definite bottom. A defined top and a defined bottom. They have polarity. The top can be called the apical surface or the free surface, and the bottom the basal surface. Okay? Some epithelial cells have microvilli. You may recall I spoke of microvilli in the cell chapter, and we said that they increase surface area and that the core of it has a microfilament actin. And then we spoke of cilia. Cilia move stuff over the top of the cell. Their core was microtubules. So some epithelial cells have a modified surface. Okay? But they have polarity. Epithelial cells are connected to one another. Remember, that's the reason I said there was nothing in between. And they are connected by something called intercellular junctions. Intercellular junctions. So now let's digress for a moment and talk about intercellular junctions. These are the things that anchor cells together. I'm going to say that there are three classes of intercellular junctions. Anchoring junctions, tight junctions, and gap junctions. Anchoring junctions merely hold cells together in their relative position. Merely hold cells together in their relative position, so they stay in a relative position. Tight junctions keep most water-soluble stuff from passing between the cells. Now, you can't keep lipid-soluble stuff. Lipids, as you may recall in earlier discussions, go straight through cells because most of the membrane is lipid. But water can be held out, and water-soluble substances. That's the purpose of tight junctions. Gap junctions allow water-soluble substances to pass from cell A to cell B with little resistance. Gap junctions allow water-soluble substances to pass from cell A to cell B with little resistance, and we'll see. So these are the three major classes. Now, under the anchoring junctions, there are two types, the adherens junction and the desmosome. So we're going to go next. The anchoring junctions hold cells in relative position. An adhering junction is a junction who has actin hold it together. Now, the adherens junction, I want you to think about areas of high abrasion versus low abrasion. I'm holding the cells together. It's kind of like a needle and thread thing. You'll see. The adherens junction holds it together, but in areas of high abrasion, it would snap apart. So the adherens junction, though holding cells together, using these proteins here, is a weaker junction to hold things together, but it is still an anchoring junction. So it's the weaker type. A macula zonular. A macula means spot. So this would be like, let's say we took blocks when you were a little kid, blocks, and we put them down, and we wanted to hook one block to another block, a sheet of blocks. Then a macula would be if we took a double-sided tape and put a spot on one side, a spot on the other, a spot on the other, a spot on the other, and then put the neighboring block, so you'd be held together by spots. A zonula would be if we took the tape and surrounded the double-sided tape, so it's completely surrounded, and that forms a zone. That's the difference between spot and zonula. But the adherence junction is a weaker type. The desmosome is the strongest anchoring junction that you can have. It's stronger than the adherence. It uses stronger proteins. Stronger proteins. Right here. So in areas of high abrasion, you want to use a desmosome. Very strong anchor. This is what I call, right here would be kind of a button and thread. Kind of a button and thread. Okay. So of the anchoring junctions, you had two types, the weaker one and the stronger one. Okay. We go further. A tight junction, which also is called an occludence. What does occlude mean? To close. Will prevent water-soluble substance from passing between the cells. Sometimes it may permit a little bit, may make it a little leaky. For example, the blood-brain barrier, when we talk about it later in this course. Okay. The way this works is you interlock right here. You interlock. So the outer layer, remember, is a phospholipid bilayer. So the phospholipids interchange. The phospholipids interchange that head of them to kind of give spots where this is occluded. This is lipid right in here. So that lipid would prevent water-soluble substances from going between. The tight junction, occluding junction. Okay. The gap junction, or Nexus Junction, allows water-soluble stuff like ions to pass freely between the cells. Pass freely. Here would be an example of that. And actually what happens is you have these six transmembrane integral proteins here. And here's another cell. So that's one cell, this is another. So remember, the cell membrane is a fluid mosaic. So this iceberg of proteins, transmembrane integral proteins, would float around and form a circle. All the way through. Then in the other cell, these would float around and form a circle. And they would dock. Let me tell you what it's like. I don't know how many of you know about the space station. But when they bring supplies to the space station, you have to dock the two ends perfectly. So that nothing can get between the two. So you form a seal. It's the same thing here. You're forming a seal, and therefore water-soluble stuff and water can go between there from one cell to the other. And then you can remove it because these can disperse, just like with the space shuttle and bringing a spaceship up. Okay? But the purpose is to allow it to go. They also call a connexon junction. The two connexons fuse to form a pore between the two cells. Okay. Here again would be an example of that I'm showing you. That's one cell membrane and that's the other. See, this forms that hydrophilic channel where water and water-soluble stuff can go between. Here's it closed. It can close and then open. Okay. So what were the characteristics? Number one, cells have polarity, definite top and bottom. Second, they are attached to one another by intercellular junctions. Third thing, epithelial tissue cells rest on a basal lamina or basement membrane. Now, a basal lamina is a layer of extracellular matrix. Some books call it a basement membrane, but it's actually two separate things. And what a basal lamina is a thin layer of extracellular substances that the epithelial cell rests on. So you have carpet. The carpet shouldn't rest directly on the cement. You should have an underpinning. So I'm saying that all epithelial cells will rest on something. And what they will rest on is either a basal lamina, which is thin, or a basement membrane. A basement membrane is thicker. It's thicker because it has a couple of layers. A basal lamina is one layer. In areas where you're not having much penetration, a basal lamina. But in areas of friction and a lot of penetration, like in the skin, you need something thicker so it doesn't go through, doesn't poke through. So a basement membrane can be formed by two basal lamina stacking on top of one another, or by a basal lamina stacking on top of a reticular lamina. This is the strongest, a basal lamina on top of a reticular, which we'll see in the skin. Okay. The basal lamina is secreted by the epithelial cells above. The reticular lamina, if you have one, is secreted by the cells below, which would be in the connective tissue. Here an example right here of a basement membrane. See, here's the epithelial cells resting on this basement membrane here. Okay. So all epithelial tissue rests on a basal lamina or basement membrane. Okay. Now, avascular. Because the cells are stuck together, you can't get any blood vessels in between. So that means epithelia is avascular, like the skin. Suppose you had blood vessels in the top of the skin. The minute you rub it, it would start to bleed. So how does it get its nutrients and oxygen? It diffuses from below, from the connective tissue. It diffuses from below. That's how the cells live. But however, epithelial tissue sheds very quickly because the cells are constantly having to be formed because you're flicking them off. Flicking them off. So let's go. Avascular, but innervated, meaning you can get a few nerves in there. High rate of regeneration. High rate of mitosis because they're the top layer, so they're constantly being flicked off. They're the top layer, but they're constantly being flicked off. So they have a very fast mitosis rate. Very fast mitosis. Remember in chemotherapy, we mentioned that cells that mitose fast would be impeded by chemo. That's why epithelial tissue gets very thin when a person is a side effect sometimes of chemotherapy. Okay? So now we've talked about the epithelial characteristics. Okay? Now we're going to start a new video, and we're looking now at classification of epithelia. Classification of epithelia. Starting a new video. Okay?